So good morning, everyone, and welcome to the sixth in the series of webinars from the Industrial Transformation Research Hub in Biocatalyst for Pharma. As you all know, my name's Elizabeth Gillam, and I've got the pleasure of coordinating and introducing these webinars. Before we start, I just wanted to mention a little bit about the Research Hub and these webinars. The Hub is a group at the moment of academic researchers and a partner investigator, or a couple of partner investigators, a partner organisation from industry. We all have complementary expertise, which we plan to apply to problems associated with the use of enzymes as biocatalysts in pharma. So the point of these webinars is really to try and engage partners from industry to help us leverage governmental and other funding to support our research on projects that are relevant to and designed in collaboration with pharma. So the emphasis in these webinars is on presenting some of the science that's led to the formation of this research hub. And through the webinars, we aim to introduce you to some of the members of the hub and to demonstrate the expertise that they bring, as well as to illustrate some of the projects we could pursue in the hub. These webinars will also, though, serve as a model for what to expect from the regular forums that we will use to communicate outcomes from the hub to ensure that participants have timely access to information and tools resulting from the research underway in the hub. We've got a number of seminars coming up in the next few weeks, the schedule for which is shown in the next slide. We're over halfway through the series now, but if you missed any webinars to date, we have a YouTube channel to which the webinar recordings have been uploaded. Today, as you can see on the slide, we have another double act from Professor Phil Hugenholtz and Dr. James Behrendorff. So Phil is from the University of Queensland. He is an internationally renowned leader in the area of metagenomics. Phil is a fellow of the Australian Academy of Sciences and a Clarivate highly cited researcher. His H-index, according to Google Scholar, is 130, and he has a publication list as long as your arm, including many, many high-impact publications. But the hub doesn't include just eminent senior researchers, it also includes rising stars, and our second speaker is one such rising star. His name is Dr. James Behrendorff, and he's at the Queensland University of Technology. James has a very established career in directed evolution of enzymes, including P450s, but not limited to P450s. He spent quite a bit of time in industry, and he's worked with basically every expression system you could think of. But he today will be talking about a really neat new technology that complements very nicely what we hope to do with metagenomics in the hub. So without further ado, I'll turn it over to Phil to introduce the talk on sourcing and screening biocatalysts with novel functions from the metagenome and tell you about the workflow that we've established for efficient and focused enzyme discovery and engineering. Thanks, Phil.
Thanks, Liz. That's a very kind introduction, and thank you everybody for making time today. I just want to say a quick word about who I am. I'm a microbiologist. I'm the director of the Australian Centre for Ecogenomics, where we apply a bunch of culture-independent methods, one of which is metagenomics. We have about 50 researchers and professional staff. We have in-house sequencing capability, and we develop a lot of bioinformatic resources, including software like Genome Taxonomy Database and Check'em, which you may or may not have heard of. My colleagues for this hub will be Paul Evans, who's online with us today, and Rochelle Sue. Okay, so if you look at the tree of life and you go to, say, the Open University, you see this beautiful tree, which is showing how animals and plants are related. Very pretty, but it's actually a very skewed representation of life. So all the microbial stuff, all the stuff we can't see, all the stuff that we need a microscope to see, is down here in the base of the tree. If you use DNA rather than morphology, what we can see, you get a very different view. So suddenly everything that is large and visible to us packed into one corner of one branch of this molecular view of life. And everything else outside of this little red circle is microbial. And if you think about this for a couple of minutes, it makes perfect sense, because animals and plants have been around for maybe up to 500 million years, and life on the planet is estimated to have been around for 6 times as long as that 3 billion years ago. So of course, most of life, for most of Earth's history, microbial life is the only type of life on the planet. Microbial life is also more metabolically versatile than plants and animals. So actually, animals in particular are fairly metabolically restricted. We use oxygen as our terminal electron acceptor. We're heterotrophs. We break down fixed carbon already. Microorganisms pretty much handle every conceivable electron acceptor and donor. They harbor a lot of very interesting secondary metabolism. And actually, where you find secondary metabolism in plants or animals, you'll usually find out that it's been obtained from microorganisms in the first place. So you can think of plants, photosynthesis, that is a microbial origin and cyanobacteria, which is co-opted into plant cells. Not only are they more metabolically versatile, they can survive at all sorts of extreme conditions, which animals and plants cannot. So here are a few pictures of some extreme environments. On the left, we're seeing a hot spring in Yellowstone National Park. The water is boiling in here, and you will only find microbial life, predominantly bacterial or archaeal. In the middle, we are looking at acid mine drainage runoff from pyrite oxidation, which results in a stream of fluid that's about pH 1. And the microbial life is absolutely thriving under these conditions. And these are actually pink biofilms growing at the margins of this highly acidic material. And on the right, we have the very famous Lake Hillier in Western Australia. And the pink color is due to microbial life producing that pigment. And so this is not only saline, it's also very alkaline. And so that, I know that you guys are interested in looking for halogenases. There would be some pretty interesting halogenases within this type of habitat. So hopefully that convinces you that if you want to get novel biocatalysts, you should be looking in the microbial world. And so the issue with that up until relatively recently is that the organisms, if we want to look at the microbial world, we've grown them on an agar plate. And if you look at the cells under a microscope and you do a quantitative comparison, there's usually 2 to 3 orders of magnitude difference between the number of colonies you get out of cells and the number of cells under the microscope. So this is recognized famously as the Great Plate Count Anomaly in the 1980s. And so it's telling us that most of the microbial world has not been obtained in pure culture. So what does this mean if you're searching for biocatalysts? And I'll get onto that in a minute. So the way that this issue was overcome was through sequence-based methods, and this is metagenomics. So without giving you a very long lecture, I'm just going to cut to the chase. If you take an isolate, in this case, this is E. coli, but I could take a cheek swab from you guys and sequence your genome, that's genomics. If you take an environmental sample and you extract all the DNA from all the multiple organisms that live in this ecosystem, then that's metagenomics. So in a little bit more detail, in the laboratory, you take your sample, whatever it is, and it can be anything that has microbial life. You extract the DNA, you sequence that DNA, and then you do a number of things to the DNA. Typically, you would assemble it and you would annotate it. You would identify protein coding genes, for example, which you would do in standard genomics. There's an extra step that you do in metagenomics because it's comprised of numerous microorganisms. You want to bin out those different populations so you know which genes go with which organisms. There is an issue with this approach, and that is that the more complex the microbial community you're looking at, the progressively you get less assembly. So this is an indication of species complexity. So here's that acid mine drainage ecosystem I was telling you about. It was selected because it has very low diversity. There's only about a dozen species in this environment. Surface ocean waters are a bit more complex, in hundreds of different species. And then if you go to a soil sample, you're now getting into the thousands to tens of thousands of different species. And what this looks like when you sequence it is that you get progressively less assembly, to the point in soil, for instance, that mostly you're just getting individual reeds, which is sometimes just called shrapnel. This has been addressed most effectively with technical improvements in sequencing, associated with also with improvements in computing power. So there's a very nice tracking of metagenomics with this improvement. So up until the mid-2000s, Sanger sequencing was the dominant method that was developed by Fred Sanger in the 1970s. And there were some modest improvements like going from gels to capillary sequencing. And I used to work at the Joint Genome Institute in California, and it's very typical that you would have a room full of these Sanger sequences producing what we thought at the time was a lot of sequence data. That all changed in the mid-2000s with the development of so-called next-gen sequencing, which are all united by the fact that they're miniaturized and you do massively parallel sequencing. And this is a real game changer. So this is showing on the y-axis the amount of kilobases you're getting per day from a machine. And I'll also just point out that this figure is quite a little bit old now, and this has continued to go up, and it's a log scale. So we're getting vast improvements in the amount of sequence data. And the current industry leaders in sequencing technology, arguably, because some other providers may argue this, Illumina, which a single run on an instrument like this would produce 3 terabases of data, and more recently Nanopore, which is well known because it produces very long reads. And these both use different sequencing technologies than the original Sanger. So you can think of metagenomics in 2 phases. So before the advent of next-gen sequencing, where we were producing only millions of base pairs of data, you were often left with unassembled data that you then annotate, and you use the so-called gene-centric analysis. We still use gene-centric analysis, but it's considered generally cruder because it provides you less contextual data about the metabolic functions that you're interested in. As of the 2010s forward, we're now producing billions of bases. This allows assembly and then actually reconstruction of near complete full genomes, and they're called MAGs for short, or metagenome assembled genomes. Essentially, they're a composite of individual populations within the community. Once you have those genomes, you can then do a standard annotation and metabolic reconstruction and find out what these organisms are doing. And most of these organisms have not been cultured. So a lot of what we know about microbiology today is based on the ability to get the genetic blueprints directly from environmental samples and work out what's going on. So my group has been involved in metagenomics over the past 20 years. So we've seen it evolve over that time, beginning with the first metagenomic study in 2004 in Jill Banfield's group, Gene Tyson, who's here, was at the center here, and myself. And we, this was that pink biofilm from the acid mine drainage, selected because it was a simple community. And that demonstrated that the approach was viable. And from that, we could get the blueprints of these organisms in the acid mine drainage, including enough information that we could then culture an organism from that community based on the metabolic reconstruction. And then there's been a series of papers over the years, each of them contributing new knowledge to ecosystems that we knew not a great deal about. One of them is a wastewater treatment, very important process of removing phosphorus, which has essentially been a black box process since it was innovated about 100 years ago. And in fact, microbiologists were looking in the wrong direction for several decades, looking at an organism that could grow that wasn't actually responsible for the phenotype of phosphorus removal. We looked at termites. Termites, we were interested in the enzymes that could break down lignocellulose for biofuel production. And what often happens in these studies is you just get a huge amount of potential sequences that could be investigated and characterized. We looked at the microbiome associated with the root of the model plant Arabidopsis thaliana, and we also identified the organism, which was an archaean, responsible for anaerobic oxidation of methane coupled to nitrate. So we knew that this function could occur, but they had, from cultivation, we didn't know what organism was capable of it, and we could work out the molecular underpinnings of that. And recently, through a collaboration with Chinese collaborators, we produce large genome and gene catalogs from glacier microbiome, so cold-adapted environments. So this is just a few. Pretty much any ecosystem that you can extract DNA from, you can do metagenomic analysis. And across these 20 years, we've seen a very strong evolution of the technology. And our center has been very contributing a lot of bioinformatic tools and improvements to the way that we analyze these data sets. I'll turn my attention now to a specific recent metagenomic analysis with Neven Karagula and involving Liz and Julian Zaug, looking at extreme ecosystems in Turkey. So we looked at a couple of thermophilic ecosystems, an acidic ecosystem, and a few halophilic ecosystems, with the idea specifically, although there are a lot of other enzymes and interesting metabolism in these ecosystems, specifically look at for P450s. And so the approach was to take the reads, as we've talked about, assemble the reads into contigs and scaffolds, to bin those into the metagenome assembled genomes, do the annotation, predict the protein coding genes. And so we were getting very large numbers, as you'd expect, from each of these extreme ecosystems. And then from that, to pull out the P450s. And based on this, we heavily extended the tree, the P450 tree, revealing 76 previously unknown P450 families. So it just shows the huge potential of metagenomics for enzyme discovery. So in fact, the real issue is not the enzyme discovery part, but the way that you then take that information and process it down. And that's part of what this hub is all about and how you process, and you'll hear a bit of that from James shortly. Unspecific peroxygenases, UPOs, I know you are also interested in. So versatile multifunction protein family, the ideal biocatalysts. And the focus has been very much on UPOs from fungi, although now a few patents out there I see for bacterial UPOs. That seems largely unexplored. I'll just point out that the UPO database or base, which is reporting a huge variety of UPOs, is really 1,948 UPOs mined from 800 publicly available fungal genomes. This is a drop in the ocean. So if you look at the fungal genomes available in the public databases, these are increasing rapidly. They're increasing rapidly because of the improvements in the sequencing technology and all of the associated informatic tools. And interestingly now, MAGs are starting to appear. So fungal genomes are larger than bacteria and archaea, and so MAGs have been a challenge, but now with the long-read sequencing technology, it's possible now to recover uncultured fungi directly from environments. And that's exciting because, like bacteria, most of what we can culture in the fungal kingdom is just a small amount of total diversity within the fungal kingdom. So we can expect that we will have many more UPOs, but also very many more novel or diverse sequences from these genomes. And then moving from UPOs to UFOs, unidentified functional objects, there are greater than 235 million hypothetical proteins. So we know that they're proteins because we see start and stop codons and various other regulatory elements, but we don't know their function. This is very common, particularly in microbial genomes. Typically, northwards of 50% of the genes within within novel bacteria, for instance, don't have known functions. That represents over 2,000 conserved domains of unknown function. So this is obviously an enormous reservoir of novel functionality, some of which may be of industrial relevance. So functional characterization of hypothetical proteins has traditionally been time-consuming and costly. So now we're getting methods to increase lab-based characterization, and you'll hear that from James. But on top of that, now we have much more efficient ways of doing decreasing the functional search space bioinformatically, aided by machine learning and other artificial intelligence. And that was David's talk a few weeks ago. And so you can use these features to really refine your search space. And sometimes you may be looking at 100,000, 200,000 candidates, and you really need to reduce that before you move to the bench. So I'll just now zoom out back to the pipeline, just to remind you that where the metagenomics fits in is to basically generate your list of candidate enzymes, and then you can use deep learning that David has talked about a couple of weeks ago to refine that informatically, and then you can move into characterization and cell-free systems, as James will now describe to you.
Thanks very much, Phil, and thanks, Liz, and thank you, everyone, for joining us. So, no matter how good the annotation tools are at the moment, at least, every candidate enzyme that you can pick out of the database is still a hypothesis. Will it do what you want it to do? And as we all know, there's still only one way to really find out, and that's to try and see if you can squeeze some activity out of it. And this is time consuming and it's costly, and the costs and time expand with the number of items that you want to screen. Even if you want to use if you have a high throughput screening assay available, where you can look at a lot of variants in multi-well plate format, there's still a lot of steps and weeks taken up, usually with cloning, transformation, colony picking, validating that at least there's protein expression or getting some kind of signal out of your assay. And then if you're on detailed characterization, obviously it gets even more time consuming. And so these costs really determine and limit the scope of any of these screening campaigns. And this is really a critical point until we get to the point where machine learning can really give us much more accurate functional predictions from sequence data. In my group, we're trying to shorten this timeline and reduce the cost by eliminating all the in vivo steps. We want to be able to go from DNA sequence to synthesis of an assayable protein sample in a few hours rather than days to weeks. And the technology we're using for that is cell-free protein expression. So the system that we use, for example, we can take a DNA sample. We don't need any like prior whole cell steps. We could even get this DNA sample directly from a PCR or a commercial synthesis house, and add the DNA sample into a 10 microliter reaction volume, incubate it for 2 hours, and have protein after those 2 hours that's ready for assay. This tiny volume allows us to screen dozens to thousands of coding sequences in parallel. And if there are high throughput screening assays available, then screening can be completed in a single day. It does sound almost too good to be true. And if you've had prior direct experience with cell-free protein expression, you might already be gritting your teeth. So let's go back to the basics of what cell-free protein expression is about. It's everything that you need for transcription and translation in one pot. So amino-acylated tRNAs, energy regeneration cofactors, some form of RNA polymerase, obviously a lot of ribosomes, but no resources in that reaction are being directed to cell replication or other functions that we're not interested in besides protein synthesis, which sounds great if it's working well. If it's not working well, then the rest of that cellular support that makes protein synthesis function and replenish parts that are breaking, the rest of that resource isn't there. So there is some work that goes into making a high quality cell-free protein synthesis system. How do you make it? It's basically any whole cell extract. You could make a cell-free translational extract from any living organism. The oldest and original cell-free expression system was made from wheat germ, and it's still used a lot today. It makes very high quality proteins at extremely low yields. HeLa cells are also a common source, along with rabbit reticulocytes. Saccharomyces is not used so often along with plant cells. Probably the most widely available cell-free system, both commercially and being made in people's labs, is from humble E. coli. So you make an extract of your live, viable, ideally rapidly growing cells. You fractionate this extract, go through a few desalting columns, and then supplement it again with anything extra that might have gone missing during the purification. Additional nucleotides, amino acids, sometimes more tRNAs. And something to note on here is that the batch-to-batch variability in preparations of cell-free expression extract is very high, and this is globally recognized as a challenge. Not many groups have really taken a systems biology lens to the preparation of cell-free protein expression extract as a reagent, and that's a big reason for the batch-to-batch variability. So it's important to have a lot of validation assays that allow you to interrogate that the different parts of the system are functioning correctly, and you're not just relying on your favorite protein dropping out of the end of the pipe. Our preferred organism for making cell-free expression systems is this guy here, Leishmania tarantuli. So it's not pathogenic to humans. It's a pathogen of lizards, but it's a single-cell eukaryote that we can easily grow in a bioreactor. So it's a eukaryote with some of the charms of a prokaryote for being able to work with it easily in the lab and grow it up to high densities. Why do we choose Leishmania? It's a question of balancing the complexity of the system with protein folding needs. It's attractive, the idea of having the simplest, most defined cell-free expression system possible. For example, the Pure system, which was developed by Mike Jewett and is sold by NEB, is reconstituted completely from purified components, just exactly what's required in there. I think it has 23 ingredients. It's got purified ribosomes, tRNAs, etc. It's very predictable in its performance in terms of like how much polypeptide it's going to synthesize. But those polypeptides need to be able to fold completely unassisted. It's only really good for the most stable, easy folding proteins that you can think of. Not really much besides GFP. And it's also more expensive than gold. So going up a little bit in the complexity of the system, it's much more common to make an E. coli-based cell-free protein system. These are also available from several retailers, and a lot more people prepare E. coli-based system in their own labs. It's cheap and simple to prepare, and it is great at making a lot of polypeptide very fast, but it's terrible at folding complex proteins. It's like if you think back to the problem of like if you're overexpressing proteins in E. coli, if you've got very high rates of transcription and translation, but your protein of interest has a lot of hydrophobic regions or challenges in its folding, you can quickly end up with a lot of aggregate. And all the old techniques about like reducing temperature to slow down the translation rate all speak to the challenges that people have with E. coli-based cell-free protein synthesis. So again, you can make a lot of polypeptide, but in terms of what our interests are, that's not helpful if the proteins aren't folding and functional. And so the Leishmania system, its rate of translation is much slower than E. coli, and the total yield is also much lower, but it has a vastly superior rate of successful folding. The proportion of proteins that are correctly folded in the Leishmania system is so high that even with the lower total yield, you get much more assayable material than when you're expressing with E. coli. Another benefit of the way we prepare the Leishmania system is it retains a lot of microsomal membranes, and we can get spontaneous membrane insertion of a lot of membrane proteins, both membrane-associated enzymes like P450s and membrane integral proteins. There are a few papers now demonstrating that these proteins actually pop into the microsomes and are functional. We've got a lot of experience with this system. Within my group, there's more than 10 years collective experience with Leishmania cell-free, and we're on the same campus as Kirill Alexandrov's group, and Kirill's group initially developed Leishmania as a cell-free expression system and actually commercialized it through Jena Biosciences. And if you ever buy the system from Jena Biosciences, it's actually been made in the lab next door to my office here and shipped on a regular basis. Fortunately, we just get it fresh out of the tap, let's say. We have also recently developed a lyophilization strategy. This is the first eukaryotic cell-free expression system that's been lyophilized, so we can actually ship it at room temperature now. Okay, so moving on to what we actually do with it, it's very good for expressing highly stable enzymes. So in this example slide here, we've got a thermostable esterase and a thermostable bacterial lacase. And in a 10 microliter reaction, we actually have to dilute the protein expression reaction product. We have to dilute it substantially to be able to get close to linear reaction rates in a colorimetric assay. So for me, this was pretty exciting the first time I did this, to be able to, in 2 hours, pop in some DNA and have enough material afterwards for several hundred assays. It really speaks to how you can use this system to quickly, for example, optimize conditions. You can try all the different, buffer parameters, temperatures, pHs, anything else, cofactor additions. From just one reaction, you've got a lot of scope for answering a lot of questions very quickly. But importantly, it does also work for trickier enzymes. We developed expression of cytochromes P450 and P450 reductases. Initially, we developed these independently. There was a little bit of optimization needed, particularly when it came to how and when to add the various cofactors into the system. But we could easily and quickly work through this process with high throughput assays. We could use the cytochrome C reduction assay for figuring out the right conditions for the P450 reductase. And in collaboration with Liz's group, we used reactive oxygen surrogates to provide electrons to the P450 for being able to optimize its expression independent of the reductase. And then we put the two pieces together and reconstituted a complete P450 reductase system. They do have to be expressed together in the same tube, which suggests that they do need to insert into the same microsomal membrane together. You can't express them in separate tubes and then try to combine them. And then with the reconstituted system, this is an example that we had with an industry partner who had a natural product that they wanted to make that relied on a challenging P450 system that had previously only ever been expressed in fungi. Using the Leishmania cell-free system, we were quite readily able to express this particular P450 and its cognate reductase and cycle through a variety of modifications of the P450 to try and engineer it for expression in a simple prokaryotic host, which was successful. And again, at these very low scales, we were able to produce sufficient functional enzyme to go directly into enzyme assays and validate product formation with LC-MS. We can also use the system with all the classic human P450 systems. So we reached for CYP2D6 as an example of an important human P450 that's not necessarily straightforward to express in E. coli. Of course, it's been done, but we were able to express and assay the native 2D6 sequence with no N-terminal modifications whatsoever. We can assay these enzymes with the P450 glow substrates from Promega. We can do inhibition curves. I've shown a couple of example curves here. I quite like that we were able to, so as 2D6 and also some of the other more challenging enzymes we've worked with, the fact that we've been able to assay them without any modifications is very encouraging to me, that we'll be able to use the system for more and more agnostic screening, where we're just taking sequences, putting them in and trying to get outcomes, and not having to retrofit sequences to fit the expression system, but rather having an expression system that's ready to screen what we throw at it. And important to note here, we did try our best to express these enzymes in E. coli-based cell-free, just to kind of see whether we weren't just working with our favorite system for the sake of it, but with E. coli-based cell-free system that we made in-house and also purchased commercially, and we couldn't get anything out of it. So what else can we do with it? I think this can be a really valuable tool for speaking to the idea of improving annotation quality through machine learning. We can go from a metagenome sample to sequencing and annotation, then take those annotations and go directly into cell-free expression, either from by PCRing up a sequence of interest from the original DNA sample or synthesizing it commercially, proceeding to functional characterization, and then feeding that data back into the annotation. It really fits with the machine learning paradigm that the key is that we can express enough enzymes and do enough assays that we can produce sufficiently voluminous and high quality data for machine learning to actually work. On a similar note, we want to use this system to better understand the determinants of reaction selectivity. So this is particularly a P450 question as well. You can have closely related enzymes that have subtly different regioselectivity. For example, on a substrate, you might have hydroxylation at one position or at the position next to it, and this might be really important to you, and you can't really pick it out just from sequence data. If we can functionally screen large numbers of enzymes and go directly into even qualitative LCMS and feed that data back into machine learning framework, we can hopefully be able to go from sequence to functional prediction. The system is not good just for looking at individual enzymes. We can reconstitute entire pathways. This little cloud down in the bottom left corner of the screen, some of the enzyme families that we've worked with a lot now, and we have confidence that we can work with these systems routinely. P450s are not the only membrane-bound system in there. We've been working with membrane-bound phenyltransferases as well. So again, in this small scale format, getting out proteins, we can express multiple proteins in different tubes and have a combinatorial style assay in multi-well plates, or we can even pull a bunch of sequences into a single tube. But you can easily imagine how you can quickly use the system for optimizing gene combinations for getting your best product yield in the end. And we've developed 4 new pathways, new metabolic pathways for reaching known natural products, and produced a couple of new to nature compounds. And again, regrettably, I can't go into the details of that because those are all industry projects. We can potentially do even more with this system. Even though we're dealing with a lysate made from a eukaryote, there aren't any cell-free expression systems that can readily do protein glycosylation, because the transport network and the organelles for protein glycosylation are obviously destroyed when the cells are homogenized. So we're looking into building up a project where we integrate simplified protein glycosylation systems from bacteria. There are a few N and O protein glycosyl transferases from streptomycetes that we're interested in testing here, where we could have non-native glycosylation, but sufficient simple glycosylation to still aid protein folding. And we've got some good bacterial and fungal lacases in-house that we can use as a model here. We've got a bacterial lacase that works extremely well in this cell-free system, and a fungal lacase which we know is hyperglycosylated and does not work in our cell-free system so far, but we can produce it readily in the yeast as a control. And so we've got a good model system for developing glycosylation, and that's something that we're really keen to do. And it would be very relevant to the UPO work that's been proposed here for this hub as well. Further into the future, the system is used here on campus for mRNA display, for panning enormous libraries of binding domains, for finding protein domains that bind to specific ligands. With mRNA display and cell-free protein expression, you can functionally screen libraries greater than 10 to the 12 individual variants in a volume of about 10 milliliters. And we're brainstorming and exploring how to adapt this to potentially an in vitro based screening and evolution opportunity for biocatalysts. And with that, I'll hand back over to Liz.
Thanks, James. So thanks, James, and also thanks, Phil, for showing what the capabilities are of these sort of twin technologies. I will be editing out any questions from the recording for this this session, but I'd like to open it up now for questions from any of the people online. So please feel free to turn your microphones on and ask some questions.